Is there a very clear and provable paper trail that proves that the US government did in fact work to help manipulate and create this latest coronavirus, and that this virus is actually the product not of nature, but of man-made manipulation? The answer to that question is yes.
Many of the things that have been postulated to be novel about the coronavirus that we're currently talking about are things that were very clearly documented in experimentation in vitro in labs going back as far as 2013 in patent applications and in grant filings.
Hey guys, welcome to the show, and today I'm speaking one-on-one with Dr. David Martin. You've probably seen him in Plandemic 2, and today we're talking in great detail about something I've been reporting about for 2 months now. The very clear paper trail that connects US labs, DARPA, and the creation of a manipulated coronavirus over the past couple of decades that has ultimately led us to where we are today. Now, fact checkers will tell you all of this is conspiracy theories, but here's the problem. It's not based in theory at all. It's based in factual source documentation, black and white paper trail that's very clear. Before we get into all of our interview today, I want to give a shout out to our sponsor for this episode, ise.media. If you have not checked out our new platform yet, please do. It is a platform for content creators who are being purged and removed from social media sites like Facebook and YouTube. At ise.media, you're going to get the uncensored truth, and all of it, again, is factual, fact-based, and sourced information, unlike what the fact checkers are telling you. And by the way, we're running an equity crowdfunding campaign right now. You can actually own a piece of this network, help us to build it, help us to reach our funding goal. We are growing every single day with more than 1,000 individual investors now in the company. Be a part of history, ise.media. Well, Dr. David Martin, thanks so much for being with me today, and I want to begin by talking about this issue that I have actually been researching now for over 2 months, and that is the paper trail that proves that the US did in fact work to create a manipulated coronavirus, outsourced to labs in China, 6 different virology labs, and paid for development there, including the lab in Wuhan. The documentation that I have found so far, I have reported on, but you are now providing new information on this story. What can you tell us?
This is a great conversation, and one that really deserves an entire expose, because the fact of the matter is the NIH made the determination to stop gain-of-function research. And as we show in the letter that was sent to funded organizations, the one we made reference to is the one that was sent to the University of North Carolina at Chapel Hill, specifically directed at the coronavirus architect of chimeric mutations of coronavirus, Dr. Ralph Baric. And as we showed in the film, what the letter said was that the NIH had concluded that the work that was being done potentially was problematic. At the bottom of the letter, it basically said, since your grant is already funded and ongoing, we're not telling you to stop. Co-funded grants, funded both by the Chinese government and by the United States Department of Health and Human Services through NIAID, Anthony Fauci's organization, and through the National Institutes of Health generally. Now, here's the tiny problem. We, as a nation, in 2012, going into 2013, decided that the work that was being done crossed an ethical and potentially legal line, and we as a nation stated we weren't going to advance it. Every one of the letters that was sent to every funded researcher, both domestic and foreign, all came out with the same statement that said, you're not supposed to do the research, but ... This is a recognition that the National Institutes of Health and the National Institute for Allergies and Infectious Disease, Anthony Fauci's own organization, specifically took the will of the people and suborned it without question in writing in these letters that essentially said, keep funding. And that's where we have a tiny bigger problem, because that tiny bigger problem is that many of the grants that were going to the active chimeric coronavirus researchers, many of those grants were ongoing and evergreening grants. There's a thing called an R01, a center grant, which is one of the instruments through which the National Institutes of Health and National Science Foundation and others get funding to researchers. In many instances, these evergreening grants were ones that effectively, based on the NIH's own written statement, said, while we're going to tell you that we think your research is problematic and potentially unethical or illegal, we're going to go ahead and look the other way and allow you to do it. And that included funding graduate students who also themselves had joint appointments both in the US and China.
Yes, and there is no question about that, but there is another question on top of that, which goes back, it's almost the precursor question. The why. Why was this deemed to be immoral? Why was this deemed to be possibly illegal? And from what I have seen of documents at that time, there was an honest debate going on within the scientific community of whether or not the essentially mutation or the alteration of coronaviruses to make them attach more easily to human cells was the right or wrong thing to do. Have you found that as well? Is that what that debate was over?
Yes. In 2005, Ralph Barrett made a presentation to DARPA. This, by the way, is not only a public record, but it's on his professional CV. In that presentation and in subsequent work, there was an awareness that coronavirus represented a very interesting mutable pathogen, which would allow you to potentially build, both for offensive and defensive purposes, means by which a population could be either inoculated or infected. When DARPA gets involved, obviously the funding trail becomes much more problematic. But we do know that in 2005, the United States Army and DARPA began a process through which they started manipulating the coronavirus, specifically by taking two different strains of coronavirus and amplifying pathogenicity of the virus. Now, you don't have to be a scholar, you don't have to be a geneticist, you don't have to be a medical ethicist to realize that when you begin the process of not only in your own labs, but with international graduate students present from all over the world, when you have the ability to start showing the world how to manipulate a pathogen for potentially offensive and defensive military applications, the risks that are associated with that start going through the roof. So we have a period from 2005, when we know the DARPA initiative got underway, to 2013, when Dr. Barrick submitted in grant applications information about a super pathogen, which was his chimerically altered coronavirus. We know that during that period of time, and as you know, it's overlapping the period of time that MERS came out, which was the Middle Eastern respiratory syndrome, which most people seem to forget because it didn't really land with us here. But during that period of time, we know that a highly virulent form of the pathogen was lab created. And we know that mutation was funded by and was supported specifically both by DARPA and by the National Institutes of Health.
And it's to that point, we also know something else, which is that go back even before that. Because you're right, 2005, there was a great deal of discussion about this. But go back to 2002, 2003, and the original coronavirus, SARS, right? One of the, for lack of a better term, complaints about SARS in the medical community, and certainly from people like Ralph Barrick, was that it did not attach well to human cells. And therefore, remember, a lot of folks do not remember that SARS itself self-eradicated. There was no vaccine created to get rid of SARS. It self-eradicated in a very short amount of time. And so the complaint was that this coronavirus did not have the ability to attach well to human cells, but if it were manipulated, if the spike proteins were manipulated, it could be altered to attach better to humans and therefore could be more lethal.
Not to take you further back in the time machine, but we need to go back a little further. Remember that for the decade of the 90s, going back to the late 80s and all through the early and mid 90s, Dr. Ralph Barick's work on coronavirus had to do with rabbit cardiomyopathy. People are not familiar with that because we're not familiar with the fact that coronavirus, for the majority of the human and the mammalian evolution, has actually been more concerned of the vascular system than it has been with respect to its effect in the pulmonary system and the breathing apparatus, right? So it was in 1999, and this is important, it's 3 years before SARS, the outbreak. That 3-year period was when Dr. Ralph Barick began the mutations of coronavirus, which included focusing on the receptor binding domains, the spike proteins, and specifically the ACE2 receptor, which is what lands the virus so neatly into both the lungs and potentially into the kidneys, where we have a high prevalence of the ACE receptor in the human system. So ironically, we don't have the evidence, and the evidence would be probably impossible to source, of the degree to which the overlap that took place between the first alterations of coronavirus in the population occurred starting in funded research in 1999, but we know that from 1999 to 2002, so before SARS 1.0, we know that humans were messing with SARS specifically around those pathogenetic links. And one of the questions that I merely posit, because it is in fact just a question.
Right.
You know, we know that the researchers who were looking at this were in and out of their own labs and in and out of caves and working with bats and all sorts of other things. That's very well documented. We don't have any way of establishing whether some of the things that humans did to alter coronavirus actually went not from bats to humans, but potentially from humans to bats back to humans. I mean, the possibility, unfortunately, in the official narrative is that everything goes from an animal to a human. At no point are we willing to hold as a possibility that some of the mutations that we see in the coronavirus may in fact have gone from laboratories to nature, then back to humans. So these are things that we don't have information about, but what we do know is we can go back to 1999, 2000, and 2001, and we can look specifically at the targets that Ralph Barick and others were focused on with respect to the respiratory side of this pathogen, and then ask ourselves, how was it that we went through all of human history, had the evolution of coronavirus, and it was largely a cardiovascular problem, and suddenly we start messing with it in 1999, and 3 years later we have the first outbreak. We start doing more chimeric alterations in 2012 to 2015, and lo and behold, we start having abnormal and untested flu-like pathologies in 2017-18, 2018 and 19, and then we decide to start testing for coronavirus in 2019 going into 2020. The fact is, we don't have pathogenic data to show us whether or not SARS in a different form was around in the 2017-2018 flu season, which killed tons of people here in the US and around the world. We don't know.